Have a nice and safe day to you all. A very warm welcome to this channel again. Today we are going to discuss another new topic uh, that is about porifera, the general features of porifera. We know sponges which are variously distributed with different sizes in aquatic medium, but we have not detailedly known about the characteristics hidden behind the sponges. Let's discuss today about those interesting group of individuals which are sponges. This phylum porifera was first identified by Robert E. Grant. Robert E. Grant. Okay. First identified and named this phylum. These are popularly the porifera. Porifera deals with pore-bearing individual, porous and ferae. These are commonly called as pore-bearing individuals. Okay, as we know, this porifera is the most primitive phylum in animal kingdom. It is most primitive phylum, having less number of individuals, most primitive in animal kingdom with very less number of individuals. These are aquatic in nature, having both fresh water and marine water. These are mostly found in marine water, while a very less in freshwater habitat. Okay, these are sessile. There is no locomotion power for them. Okay, suppose we'll take a structure to determine its components. It shows that the body is vase-like this. The body is vase-like, diploblastic bodywork, outer ectoderm, or it is pinacoderm, inner gastroderm or guanoderm. This cavity is spongiocele. The body consists of a number of very small openings are there. These openings are ostium. Okay, the openings are ostium. The ostium open into a central cavity. The central cavity is spongiocele cavity. Spongiocele. Okay. The terminal opening is osculum, or it is called as mouth of the sponge. The outer layer is pinacoderm, which consists of pinacocyte cells. Outer layer is consists of pinacocyte cells. Like this, suppose these are pinacocyte cells. In between pinacocyte cells, there are openings. The openings, these are pinacocyte cells, this is the opening. These openings are ostium. The opening is due to presence of another cell, that is like this. The cytoplasm of this cell bear a hole inside it, which cell is called as porocyte cell. Porocyte, porocyte cell. The porocyte cell, it maintains the ostium on its surface. Okay. The inner layer consists of it is gastrodermis, or it is pinacoderm, it is guanoderm. The guanoderm consists of flagellated cells like this. These cells are flagellated, called as guanocyte cells. Guanocyte cells are there. Along flagella there is a nucleus. This cell is guanocyte. Okay, so it is called as guanodome. The flagella beat continuously and direct the water to go out through the osculum. So a continuous canal system can be maintained inside. The in between, if we take the section of it, it shows the in between ectoderm and endoderm. These are diploplastic. Ectoderm and endoderm, there is mesogloia. This is mesogloia. The mesogloia, otherwise called as mesohyl, mesogloia, the term which is very much fit for cnidaria, but the mesogloia or mesohyl is a non-cellular layer. It consists of spicules. It consists of spicules like this: triaxon spicule, monoaxon spicule, tetraaxon spicule. The chemical composition of these spicules are calcium carbonate or silicon dioxide. Calcium carbonate, calcareous spicule, silicon dioxide, siliceous spicule. And around these spicules, some thread-like structures are found. These are thread-like structures around these spicules. These thread-like structures are called as spongin fiber. This spongin fiber, it makes a inner core material which maintain the shape of the body. Then the endoskeleton is made of spicule and spongin fiber both. Okay. The mesohyl consists of also amoeboid cells. These cells cannot remain at a particular place. These are called as amoebocyte cells, which are irregular, and a particular amoebocyte cell which is present here is called as archaeocyte cell. This archaeocyte cell is one of the totipotent cell. With among all amoebocytes, archaeocyte is the most important totipotent cell, which have important functions in internal body, in gamule formation. We have to read it in its reproduction. Okay. The inner flagellated cells, this cell, guanocyte cell, have specific function. What are the functions? Let's discuss in detail. If we see the guanocyte cell, the guanocyte cell consists in its collar. There are like this. It consists of a circle of villi-like structures. Circle of, this is the base of the cell. The upper part produces villi-like structure. Okay, these are villi-like structures. But the function of villi, all these villi help in producing mucus and trapping the food material. Mucus. So a number of villi, or this is called a collar cell, collar region. Collar having microvilli. Microvilli are present in collar. It consists of mucus which collect food particles. Food particle, when come through the canal system, it trap the food particle, engulf the food particle, and the food particles get digested inside it, and below it there is another cell. This cell is amoebocyte. Through this amoebocyte cell, the food are transported to different part of the body. So the digestion is intracellular. It takes place inside one cell. Then respiration, it takes place through the body surface. Excretion, the excretory product are ammonia, waste products. These are also removed through the canal system. Okay. The basic interesting character, the most important character, the signifying character of sponge is the presence of canal system. What is different type of canal? Suppose we will take a sponge. Here the sponge consists of canals which directly communicate into the body cavity. These are the openings directly communicate into the body cavity. It is Ascon type canal. Ascon type. When the canal is like this, like this, so this is the body wall. One canal opening closed here and another canal it open internally. This is incurrent canal, radial canal. Radial canal open into it. There is a connection between incurrent and radial canal. So water goes through this pathway. These are cyconoid, slightly complex cycon type. Then another canal which is leuconoid, highly complex. This canal, another canal, they again communicate with another canal, finally open into spongiocele like this. This sac ultimately open into the spongiocele. This is called as leucon canal, leuconoid canal. After leuconoid, there is another canal called as rhagon type of canal, rhagonoid. The rhagonoid canal consists of two lateral pouches. The lateral pouches open into spongiocele. The ostium open into lateral pouch. Ostium open into lateral pouch. Lateral pouch open into the spongiocele. This is rhagon type of canal. Okay. So the canal system is the interesting part of phylum Porifera. Okay. All about digestion, respiration, excretion. The reproduction, asexual reproduction takes place and sexual. Asexual reproduction takes place by formation of gametes. The gametes are formed by archaeocytic cell. They aggregated in the mesochloia. Suppose these are archaeocytic cell along with spicules and gamma, it produces gamete. At favorable condition, it comes out producing new sperm. This is asexual type of reproduction. Sexual reproduction, all the sponges are bisexual. They release the gametes through the water current into the surrounding water. The sperm of one sponge fuses with ovum of other, because in animals the maintenance of heterogeneity can initiate the features of race. उसमें क्या होगा ना, क्रॉसिंग ओवर होगा तो डिफरेंट गैमेट फ्यूज करेगा तो जेनेटिक फिगर अच्छा होगा. इसीलिए क्या होगा ना, जो एक एक ऑर्गेनिज्म का स्पर्म ओवम फ्यूज कर जाएगा, सेम जीनोटाइप होगा तो क्या होगा, दे बिकम जेनेटिकली वीक. So to avoid this problem, to avoid this, all animal favor cross fertilization. So cross fertilization takes place here in aquatic medium. After fertilization, what happens? A larva is formed. They are amphiblastula or parenchymola larva. After fertilization, it produces embryo and larval stage. Larval stages are found. As larval stages are found, so the development is said to be incomplete type. in so in such cases the development is indirect type of development, indirect type. Okay, so it is all about the phylum porifera. We have to describe its feature, habitat, shape, size, structure, locomotion. These are not microscopic, they are very much large, even if they extend up to 2 to 3 meter in length. Okay, so these are visible to naked eye. So all about its characters, locomotion, nutrition, respiration, excretion. It is all about the whole characteristics of sponge. Okay, we'll discuss about the canal system in the coming video. Thank you all.